So good evening, everyone. This is your third session, so let's wait for some time for others to join, then we'll just start. By the time I'll share my screen. So is my screen visible? Can someone confirm that?
Yes, it is visible.
Fine. So let's wait for 1-2 minutes, then we'll start. Okay, then let's start. If other people will join, then we will admit them eventually. So good evening, everyone. This is your third session. And in this session, we will be discussing about the week 3 topics and we will also practice few MCQs. So week 3 topics includes mutation and instability of human DNA. So by this time, you all have learned a lot of things regarding mutations because it was also covered in the week 2 as well. But in this, you'll learn about the mutations and how it leads to gain of function as well as loss of functions, and what are the different types of mutations and different polymorphisms that arise, and how it leads to instability. You'll also learn about the dynamic mutations and what are those. You will also learn about the pathogenic mutations, and in dynamic mutation, you'll learn about the repeat expansions. And then in this, you will also learn about the molecular pathology, in which there will be different types of mutations, as well as you will also learn animal models, for example, knockout mice, and why we use those models. So we will practice few MCQs related to that, and will not go into the details as it's already covered in your lectures. So now let's proceed. So firstly, I am going to discuss with you the little bit of content, and then we'll practice the MCQs. So, when we listen about mutations, then what we should remember is that mutations are the any change in the DNA sequence inside the cell, and these can be caused due to several reasons. And few reasons are due to the mistakes that occurs during the cell division, or it can also happen due to some external agents that damages the DNA. So, DNA damaging agents can also lead to mutations in the DNA sequences. So mutations are not always harmful. They can be beneficial, or sometimes they don't have any effect. And when we hear about mutations, then these are not certain processes that occurs at a particular time, but these are occurring throughout the evolution. So certain mutations are neutral, which do not have any effect on the genes or the genome, whereas certain mutations have the effect that can be both harmful as well as beneficial. So, when the mutations that occurs in the eggs or the sperms, then they are inherited. But whenever such mutations occur in the other types of cells, such as somatic mutations, then they are not inherited. So now let's see into different types of mutations. So on the basis of origin, mutations can be classified as spontaneous and induced. So spontaneous means that occurs spontaneously without the presence of any mutagen. And induced are the mutations which occurs when you provide some mutagen and that mutation is induced. So when it is induced by the external agent, then it is known as induced mutation. Then comes cell type. So as I've already discussed, there are two types of cells. One are somatic cells and one are germlines. So somatic cell mutations, they are not inherited and they occurs in the non-reproductive cells. Whereas germline mutations, they are heritable and they occurs in the reproductive cells such as egg and sperms. Then comes effect on function. So mutations can also be categorized on the effect on the functions. So one type of such mutation is loss of function, or we can say knockout or null mutation. So it happens when it eliminates the normal function of the gene. Then the mutation is referred to as loss of function mutation. Then another one is hypomorphic or leaky. That means a mutation that is reducing the normal function when it occurs in the gene. Then comes hypermorphic. So hypo and hyper. So hypo means reduces the normal function. Hyper means it increases the normal function. So that is known as hypermorphic mutations. Then comes gain of function or ectopic expression. So whenever a particular mutation leads to gain of function or which is leading to the expression of a particular gene at incorrect time or inappropriate cells, so that is known as gain of function mutation. Then comes the mutations on the basis of molecular change. So if there is a substitution in the nucleotide, then on the basis of that, mutation can be transitions. So transitions means when a purine is converting to another purine or pyrimidine is converting into another pyrimidine within a sequence of DNA. Whereas transversions, so in this, purine can convert to pyrimidine and vice versa. Pyrimidine can convert to purine. Then comes insertions. So if there is extra nucleotide is added within the sequence, then that is insertion event. And similarly, deletion means if particular nucleotide is deleted from the DNA sequence. So these are the mutations on the basis of molecular change. Then comes effect on translation. So there are certain mutations that happens in the sequence, but they do not get observed in when we look at the protein. So these are the mutations, for example, silent or synonymous mutations. So as the name suggests, silent, so means their effect is silent. That means there is a difference in the sequence of the DNA, but that DNA sequence is again coding for the similar amino acid. That's why that effect of mutation is not visible at the level of protein or in the amino acids. Then comes missense or non-synonymous mutations. So missense, that means there is some difference in the amino acid that is being coded. So it is a mutation in which earlier some amino acid was being coded by that sequence, but now due to mutation, some another amino acid will be coded. Then comes nonsense or termination kind of mutations. So as the name suggests, non-sense, which means whenever that mutation will occur, so it will lead to the formation of the stop codon and hence it will terminate the translation that was occurring by that DNA sequence. So it will lead to truncation of the protein. Then comes frame shift mutations. So whenever this mutation occurs in the sequence, so it changes the frame because we know that the whenever we convert the nucleotide sequence into the amino acids, then we take the triplet codons and then we make amino acids from them. So whenever the frame shift happens, so it means the frame will be shifted towards the right, due to which different amino acids will be coded by the entire DNA sequence. So which means shifts the triplet reading of codons out of the correct phase, means in which it was earlier read. Okay, so now let's talk about the dynamic mutations. So in this week 3, it is mainly focused on the dynamic mutations. So what are dynamic mutations? As its name suggests, dynamic. That means it is continuously occurring. And these are the mutations that are caused by the expansion of existing polymorphic DNA repeat sequences beyond a copy number threshold. So as we know that majority of our DNA is carrying the repeat sequences. Either these are satellite sequences or repeat sequences, mini satellites, microsatellites. So there are many repeats present in the DNA sequence, but whenever these repeats exceed a particular limit, then these are harmful for the cell. So dynamic mutations means whenever these repeats get expanded. And if you want to distinguish between the dynamic mutation and the other form of mutations, so the difference is that in the dynamic mutations, it is a process. It is not a single event. So that's why it got its name as dynamic. So it occurs continuously. Whereas if we talk about other mutations, then it is happening in a single nucleotide and that's it. But in dynamic, it can occur multiple times. It is a process. So, for example, here I have shown in this figure, there are different examples of the disease which occurs as a result of dynamic mutations, and these diseases are generally associated with the trinucleotide repeats. So, whenever there is expansion in the trinucleotide repeats, then these diseases occur. So, for example, in the first, you can see Fragile X syndrome. So, this particular sequence, trinucleotide CGG, whenever it expands beyond the 200, so whenever its number increases by 200, it's repeat. So then it leads to the Fragile X syndrome. And where this sequence is present, it lies in the promoter region. So this is the region when you see the RNA. So there are promoters, exons, introns. So means before the splicing has occurred, or you can consider it as a DNA sequence as well. So before the splicing, or you can say it is as a DNA. So in the promoter region, this particular CGG repeat, if it expands above 200, then it leads to Fragile X syndrome. And what effect it shows? So it leads to transcriptional silencing and loss of function mutation. So it is leading to the loss of function. And then comes Frederick ataxia. So this repeat GAA, this trinucleotide is present in the intronic region. And if this repeat expands above 200, then it leads to Frederick ataxia and it causes impaired transcription and loss of frataxin function. So it will not lead to the proper transcription of this particular DNA sequence, and it is present in the intronic region. Then comes Huntington's disease, and it is a most heard disease and most well discussed. So in this, what happens? This particular CAG trinucleotide, it is present in one of the exon, and if its repeat expands beyond 40, then it leads to Huntington's disease. If the repeat are less than 40, then it is bit means it is tolerable. But above that, it leads to this disease. And along with this, there is another disease that is cerebellar ataxia and SCA1, 2, and 3. So these are also associated with this particular trinucleotide expansion. And what happens when this trinucleotide expands? Then this trinucleotide codes for the glutamine. So glutamine amino acid. So there will be multiple glutamine synthesized as this repeat has expanded. So this what it will lead to? It will form some aggregates and it will lead to gain of function. So in this way, this is a gain of function mutation. And then comes the myotonic dystrophy. So in this, the trinucleotide involved is CTG, and it is present in the 3' UTR region of the DNA when we converted it into the RNA. So these are the untranslated regions, which won't code for the protein sequence, but they will be present in the RNA. So when these repeat expands beyond 50, then this particular disease occurs. And what are its effects? So expanded CTG repeats confers new properties on RNA, so it also leads to gain of function. So in this, we have seen that dynamic mutations can also lead to loss of function as well as gain of function. And it can occur in different regions. It can happen at the promoter or exon or intron or the untranslated regions. So in the throughout the DNA, it can occur at any region. It is not specific that it will only happen at the exon region, and it is not that means it will not only affect the protein formation, but it can also affect the transcription as well. So in this way, these are all about the dynamic mutations. So now let's see few other table. So in this, I have shown you that dynamic mutations can have different inheritance patterns. It can be autosomal dominant, it can be X-linked dominant, it can be autosomal recessive, and X-linked recessive. So these are the examples of different disorders. So first one is dent dentatorubral pallidoluysian atrophy. So this is autosomal dominant, and these are the repeats that are involved, and these are the chromosomes in which that particular sequence is present. And these are the examples. For example, this symptom onset is variable and depends on trinucleotide repeat length and can present with ataxia, dementia, depression, anxiety, chorea, etc. So most of these trinucleotide expansions or repeat expansion events, so they causes the disease that are somehow affects the neural system or you can say they are related to the neurons, or it leads to some neurological disorder. So these are somehow related to the most of the disorders are leading to the neurological disorders, or either motor neurons. So in this way, these all disorders you will see they are somehow affecting the brain. So then comes Fragile X syndrome, that is X-linked dominant. And in this, CGG is a trinucleotide, and if it expands above 200 repeats, then it leads to this disease. And different examples are what it causes is intellectual disability, epilepsy, attention deficit hyperactivity disorder, autistic-like features. So these are just for your reference. You can read it, and you will know that which are the trinucleotides involved, what are the effects, and what are their inheritance patterns. These all are important. So these all occurs due to dynamic mutations. And now let's see how these dynamic mutations happen. So what is the mechanism? So for example, here I've taken the case of Huntington's disease. So in that, CAG trinucleotide expands above, when it expands above 40, then it leads to that disease. So whenever normal DNA replication is occurring, so this repeat number is maintained. But what happens if there is a backward slippage? For example, it was earlier coding, it was straightly coding, but now due to some backward slippage, this particular part has formed a loop and it has moved out of that particular parallel, these two parallel pair of DNA strands. So this is forming a loop. So what happens? This third repeat or the third trinucleotide is binding to the second trinucleotide repeat in the another DNA strand, due to which these two are the duplicated repeats. So this will lead to further expansion of this repeat. Similarly, if this will happen again and again, then this repeat will expand. And if you know about anticipation, so in that, what happens is due to this expansion in these repeats, the onset of disease in the further generations will occur faster, because with each round of replication, more and more repeats will be expanding. So that's why that is the main reason that why in anticipation the onset increases, onset age, it means onset age is lesser, and if you look at the severity, then that increases with each generation. So what happens after this expansion? So expansion of this polyglutamine track in protein, because this CAG codes for glutamine. So due to which if multiple repeats will be present, then polyglutamines will be synthesized. And what happens if these polyglutamines synthesized? So in this they have shown the wild type protein. So if these repeats are below 35, so then it will form, the protein will fold into a correct conformation. Conformation is generally the folding of the protein, and that is very important for protein to function. So whenever it will fold properly, then it will perform normal functions. But what happens if more than 40 glutamine tracks will be present? So it will be a mutant protein and it will not be able to fold properly. So there will be altered conformation. And as it is not able to fold properly, so what will happen? Different multiple units will come of this protein and they will try to interact with one another and they will aggregate. So due to which there is multiple proteins forming the aggregation. So it is a gain of function, but still it affects the uh, human being in which it is being synthesized. So it leads to Huntington's disease. Okay, let me admit. Yeah, so this is all about the how it happens. So this is about the mechanism. So now let's learn a little bit more about the Huntington's disease. So what happens? This particular CAG trinucleotide, it is present in the short arm of the chromosome 4. So in that the sequence is present, and there is a gene named Huntington gene. So that forms the protein that is Huntington's protein. So it is a autosomal dominant inheritance pattern, and it is also means example under anticipation. So as I've told you all about the anticipation, that what anticipation means. So it leads to the caudate atrophy, loss of GABA, and in the chromosome 4, 40 plus repeats leads to its onset at the 40 years of age. And it is a neuropsychiatric and movement disorder, and sometimes it is mistaken for the alcoholism or uh, and later it appears as a Parkinsonian. So in Parkinson's also similar symptoms are observed generally. So in this way, this is all about the Huntington's disease. And in this, I have shown you that in the short arm of chromosome 4, if you can see, this is the gene which codes for this Huntington's protein. So what happens in this? During the transcription in the nucleus, this DNA strand, so there are different CAG repeats, as you can see, they are present. And with each round of replication and transcription due to the backward slippage and different mechanisms that are being involved here, so the repeat gets expanded and it leads to the Huntington's disease. If that particular repeats expand beyond 40, so it will form more and more polyglutamines and which will form the protein aggregations. So in this, I've shown you the visual representation that how it is observed inside the brain. So in this, they are showing atrophy of basal ganglia, enlargement of frontal horns of the lateral ventricles. So this is the effect on that particular part of the brain. So aggregates of the mutant, this mutant protein leads to the neuronal dysfunction and degenerations. So due to which it impacts the ability of the brain to function properly as well as to manage the movement of the body, so it affects in that way due to these uh, polyglutamine tracks, protein aggregates, and affects the neurons and their arrangement. So now let's practice few questions. So this was all about the mutations and the first part of the week 3. So these MCQs are a little bit related to week 2 topics. So first MCQ is related to the PCR. So the question is PCR can be used for which one of the following processes? So in the last session we have discussed about the PCR in detail. So I hope you might be aware of it. So the options are cloning a segment of the known coding region of the chromosome, cloning the coding region present in the processed mRNA, a mutation detection, or all of these. So you have to tell that for which one of the following process we can use PCR. So PCR is the polymerase chain reaction in which you know that if we have a small sample of DNA, then we can make its million of copies. So what do you think that what would be the correct option? So can we clone the known coding region of the chromosome or just the coding part, or we can detect the mutations also with the help of PCR? So what do you think? What would be the correct option? You can guess also if you want. Then we'll discuss what will be the right answer for this. Anybody interested? Okay, if no one wants to answer, then I'll tell you the correct answer. So correct option will be all of these. So by using PCR, you can clone any segment of the chromosome. It's not specific that you can only see the coding region. You can also amplify the known coding region of the chromosome, and you can also clone the coding region present in the processed mRNA, and you can also detect the mutations. So in this, if we talk about the reasoning, so as we know, the PCR allows the isolation of DNA fragments from the genomic DNA by selective amplification of a specific region of DNA. Hence, it can be used for a variety of applications, including genotyping, cloning, mutation detection, sequencing, microarrays, forensics, and paternity testing. So I hope you are aware that with this we can amplify a specific region depending on the primers which we use that are specific to means the regions before and after that particular sequence of interest. So in that way, we can use PCR for multiple things, for example, as we have discussed here. So now let's proceed to the next question. So the next question is, which one of the following options correctly identifies the processes given in the schematic diagram? So in this diagram, you have to tell what is being shown. So in this, they have shown you the pre-mRNA and then its splicing is happening. And then these are the two different transcripts that are formed. And then some translation is happening. And then these are the protein isoforms. So when do you observe the protein isoforms? Because splicing is a normal procedure that always happens to the pre-mRNA and then it converts to mature RNA. But what you have to see here is the two transcripts that are formed. So that is the specific feature of this type of splicing. So I hope you must have idea about this kind of splicing. So these are the options.
Alternative spice.
Okay. Someone else wants to try? Okay. Yeah, so this type of splicing is the alternative splicing. As the name suggests, alternative, so it means that alternate exons will be selected in the particular transcripts. So different exon combinations will be selected to form the transcripts, and further it will lead to different protein isoforms. Okay, so let's move to the another question. Given below are the two DNA sequences and their corresponding transcribed amino acid sequences, one for the wild type allele and the other for the mutant allele. The mutant allele shows one base pair change, which is highlighted in red, and converts the amino acid cysteine in the wild type polypeptide chain to serine. A mutation of this type is known as a. So now you have to tell these are the wild type alleles and these are the mutant alleles. And this red one is showing the mutation in the sequence. And these are the amino acids that are being coded. So you have to tell what type of mutation has happened here. So as you can
Missense
see,
ma'am.
missense, okay. Anyone else? Okay, so
Missense mutation.
yeah, right. Okay, so its correct answer will be the missense mutation. And why missense? Because in this there is change in the amino acid that is being coded. So this type of mutation is missense mutation. And it is a example of the point mutation. That means a single nucleotide is being changed. Okay, so very good. Now let's move to the another question. So this says you plan to develop a microsatellite marker from a genomic locus bearing a polymorphic repeat of CGG triplets. The sequence surrounding the repeat region is made available to you, and the same is shown in the schematic diagram given below. Which one of the options identifies the correct forward primer and reverse primer respectively, which as a pair can be used to amplify the repeat region? So in this they have given you the sequence of DNA, and in this they have shown you that these are the two trinucleotide repeats which they want to expand or they want to amplify using PCR. So you have to tell what forward primer and reverse primer should be used. And do you know these regions that are present before this repeat and after this repeat? So as I have told you that in PCR, it involves the specific region of interest amplification. So with for which we must have an idea about the region that is lying before it, so that we can use specific forward and reverse primers to specifically amplify that particular region of interest. So in this region of interest is this trinucleotide, and these are the sequences lying before and after this repeat. So you have to design the experiment in which you have to use the forward and reverse primers. So these are the sequences given for the forward and reverse primers. So you have to select one option which will help us to amplify this region of interest. So what do you
Option C
think?
ma'am.
C?
C
Option
ma'am.
C?
Yes ma'am.
Okay, so someone else wants to try?
Option D.
Okay, option D. Okay, so let's see what is the correct option. So yeah, correct is option D. So why option D? Because when you want to design the forward primer, so as the name suggests, forward primers, that means it will synthesize forward from 5 prime to 3 prime. That means from left to right. So for which strand we can use the forward primer? The strand that is in 3 prime to 5 prime, because then only it will synthesize in the forward direction. So now forward primer should have the sequence that is complementary to the leftmost region of this 3 prime to 5 prime strand. So that is GTC, ATA, CGT. So its complementary will be CAG, TAT, GCA. So this one is in the D option. Okay, so now let's see for the reverse primer. So similarly, reverse primer, as the name suggests, so it will amplify in the reverse direction. That means it will be present in the that DNA, that is the strand that is in the 5 prime to 3 prime direction. So it will start its synthesis from the rightmost part, that is from this end, which is the 3 prime end. So its sequence will be A double T, then A C G CAT. So that is in the option D. So that's why option D is the correct answer for this question. So I hope it's clear that why the option D is correct and how these sequences are derived. Okay, so if there are no doubts, then let's move to the another question. So now this is the question number 5. So it says a steroid hormone receptor X, upon binding to the steroid, activates a set of genes involved in the physiological pathway promoting muscle growth. Due to a genetic mutation, the receptor X was able to interact with a few novel targets and activate their signaling cascade. Which one of the following options correctly identify the type of mutations involved? So now you have to identify that which one of the mutations is involved. So first one is dominant mutation, second is dominant negative mutation, third one is gain of function mutation, and third one is missense mutation. So in this they are saying it very clear.
It is gain of function mutation.
Okay, someone else wants to try? Okay, only few students are answering. Okay, so it's very clear in this, they have already mentioned you that a steroid hormone receptor X, when it binds to the steroid, then it activates some set of genes that are involved in some pathway. So it's quite clear that there is some activation of function is going on. So the correct answer will be option C, that is gain of function. And why gain of function? Because in which the altered gene product possesses a new molecular function or a new pattern of gene expression. So in this, what it is doing, it is also promoting the muscle growth. It is activating a set of genes. So due to which it is a gain of function mutation. Now let's move to question number 6, which says a de novo mutation can be either somatic or germline. Which one of the following statement correctly differentiates between somatic and germline mutations? So in this, they are saying that a de novo mutation can be somatic or germline. So you have to tell which of the options correctly differentiate between the somatic mutations as well as germline mutations. So option A says a somatic mutation is heritable, whereas a germline mutation cannot be inherited. Option B says a somatic mutation cannot be inherited, whereas a germline mutation can be inherited. Option C says a somatic mutation occurs in the DNA sequence, whereas a germline mutation takes place in the mRNA sequence. Option D says a somatic mutation is deleterious, whereas a germline mutation is non-deleterious. So we have previously discussed about the somatic and germline mutations. So I think it's quite clear that what is the difference between the somatic as well as germline mutations. So you can think and answer.
Option B.
Okay, option B. Yeah, someone else wants to answer this?
Option B.
Okay. Are you getting it now? So wherever you find any confusion, you can ask me. So the correct answer will be option B because it's quite clear if a mutation is occurring in the somatic cells, then it will not be inherited. But if it occurs in the germ cells, then it will be inherited. Similarly for the somatic and germline mutations as well. So it's quite clear why option 2 is the correct. So now let's move to the another question. So given below is an agarose gel run after sequencing a DNA fragment by using the Sanger sequencing method. Which one of the following options correctly identifies the sequence being generated? So in this they have given you the agarose gel after the sequencing of DNA fragment. In this they have used Sanger sequencing method. And this is the bands that are observed. So you have to tell that what is the sequence that is being generated from this. So first is this, and this is some sequence. So you have to tell what would be the sequence. This is the negative end, this is the positive end. As we have discussed during the electrophoresis also, that DNA will always move from the negative terminal towards the positive terminal because DNA carries the negative charge. So it always get attracted towards the positive terminal. So in the electrophoresis and one another thing which you must know that during the electrophoresis, what happens is the fragments which are smaller in size will move faster, whereas the fragments which are larger in size, they will move bit slow. So on the basis of that, you can sequence it very easily by looking at the bands and the position of the bands.
Option D.
Option D. Okay, let's see what will be the correct. So someone else wants to try.
Option D.
Option D.
Yes, ma'am.
Okay. Yeah, so let's see. So the correct answer will be option D. And why option D? Because the fragment which will be present towards the positive end, they will be the means they are very smaller in length. So in this case, this A will be the fragment that is present here. So it will be the first nucleotide of the sequence, and from here to here, from positive to negative, we will sequence a fragment from 5 prime to 3 prime direction. So the sequencing will start from this end. So here you can see the A is the smallest fragment. So from A we will start our sequence. So first A, then next is C, then G, then again 3 C are present, then G, then 2A will be present, then T, then A, then again G, then again 3C are present, then A, G, A, T, T. So in this way, option D is the correct answer to this question. And this is how you analyze the gel bands that are observed after the electrophoresis. So now let's move to the another question, which says mutation in a gene causes a severe neurological disorder in the offspring of both sexes, but only when inherited through the mother and not when transmitted through the father. This property of the mutation is likely due to. So in this, they are showing that there is some mutation in the gene which is causing some neurological disorder in the offspring of both the sexes. Means this disease can occur both in the boys as well as girls, but it is only inherited through the mother and not transmitted from the father. So if a particular son is affected, then
Option
that
C.
son will not. Okay, option C. So that son will not pass it forward, and this property of mutation is likely due to.
Ma'am, it's genomic imprinting.
Okay,
Yes, ma'am. Genomic imprinting.
right. So the correct answer will be genomic imprinting and not the uniparental disomy. Although uniparental disomy is observed as a result of genomic imprinting, but this phenomena is referred to as genomic imprinting, in which, for example, you can see here the reasoning. A few genes have the property to exhibit allele-specific expression either from the maternal or paternal allele, and this process is known as genomic imprinting. Here, the gene in question is most likely silenced in the paternal allele, and hence the defective maternal allele leads to the disorder. So this silencing is as a result of this genomic imprinting, and hence this option is the correct answer to this question. Okay, now let's move to the next question. Yeah, so here is one explanation also. So in this diagram, I've shown that how the silencing is occurring both in the case of male as well as in the females. So in the right side, you can see that is the parthenogenetic or gynogenetic or maternal uniparental disomy. That is as a result, means these are the result of genomic imprinting. So in the first case, the paternal gene is silenced, so only A is getting expressed and only the maternal alleles are expressing. So in this way, double dose of gene A will be there and no gene B will be present. Similarly, in the left side, you can see that is the androgenetic or paternal uniparental disomy, in which only B gene will be expressing and A gene will not express. Only the father means copy of gene is being expressed here. So in this way, this is the paternal uniparental disomy, and these are the results of the genomic imprinting in which one of the gene gets silenced and only the gene from the one parent is getting expressed. So there is next question, which says the process marked as A in the image given below, which results in the generation of recombined chromosomes from paternal and maternal chromosomes is known as. So in this figure, they have given you the A. So what kind of recombination is happening, which is leading to these type of recombined chromosomes? So these are the alleles.
Allelic recombination.
Okay, someone else wants to track
Yes,
what?
it is allelic recombination.
Okay. So its correct answer will be option A, that is allelic recombination. So as you can see, both of these are showing the recombination, and these alleles are getting, since these are recombined in both of these chromosomes. So paternal allele is transferred to this maternal chromosome and maternal allele is transferred to this. So this is the allelic recombination, and recombination is a process by which pieces of DNA are broken and recombined to produce new combination of alleles. Crossovers result in the recombination and the exchange of genetic material between the maternal and paternal chromosomes. So as a result, offspring can have different combination of genes than their parents. So it's clear from here that what kind of recombination is this. So now let's look into the non-allelic as well as allelic recombinations. So as we have seen in the allelic recombination that in both the means one of the allele from both the parents is recombined, and means the offspring will have the different combination of alleles. But what happens in non-allelic recombination? It is a recombination when the two homologous chromosomes are not aligned in the proper manner, they are misaligned, and then they are trying to recombine or showing the crossing over and recombination. So what happens in the non-allelic recombinations? There are events which leads to the duplications of the genes or the alleles, or sometimes they leads to duplications, and in some cases they leads to the deletion of that particular gene. So for example, these are the two homologous chromosomes in which these are the two alleles that are present. So as it is misaligned and the recombination is occurring at this particular region, so in one copy of chromosome you can see, so gene A and B that was present in the topmost chromosome, so it is deleted here, whereas it is transferred to the another sister chromatid of the another homologous chromosome. So due to which there is duplication of this gene A and B. So in this way, this is the known allelic recombination in which due to misalignment of the chromosomes, the recombination is occurring at the part other than the allelic parts, which can lead to the deletions or the duplication of particular alleles. So this is all about the difference between both of them. So now let's see the another question, because the second part of the week 3 content involves the model organisms that are used to perform the studies. So these organisms are generally used to see the effect of the mutations or the silencing of genes, that how it affects the functionality. So similarly, one of the model is the mice models. There are different models which we can use. So the most commonly used is the mice model. So the question is, the knockout mice models are best suited to study. So these are the different events that we can study using the knockout mice models. So you have to select the correct one. So first one is haploinsufficiency. Second one is the loss of function mutations. Third one is the recessive disorders. And fourth one is all of these. So now you have to So now you have to tell that which of the following option will be correct. Haploinsufficiency. What can you study using this knockout mice models? So are you aware about these models, knockout mice models or other models? If you have attended the week 3 lectures, then these were covered in that. Okay, somebody is saying option 4, all of these. Okay, so now let's see what will be the correct option. So you were right. The correct answer will be all of these. So knockout mice models, as the name is, it's clear from the name, knockout. So which means simply what happens in the mice models, as mice is nearly similar to the human beings. So the gene expression is nearly similar in both of them. So that's why mice is chosen, because it's very difficult to study the expression in the human beings. It is also not safe, and it is very difficult to observe. Whereas in mice it is easier. That's why mice is chosen as a model. So what we can see, one is haploinsufficiency. Haploinsufficiency means if a single copy of gene is present, that is in the haploid form. So what happens? A single copy is not able to produce the enough protein. It is insufficient. So that is known as haploinsufficiency. But there are certain cases where even a single copy of gene is present, then also the function is conserved. It does not affect the function. So by using the mice models, we can knock out a particular gene and make it only a single copy is allowed to express. So we can see either it is sufficiently able to perform its function or not. So that is known as haploinsufficiency, that we can study using the knockout mice models. Then comes loss of function mutations. So similarly, we can mutate certain part or certain genes, and we can see now either it is able to express properly as it was expressing earlier. So with that, we can also see loss of function mutations in the knockout mice models. And then for the recessive disorders, so similarly in we can also see the recessive disorders as well. So we can study them using the knockout mice models. So in this way, the correct option will be option 4, that is all of these. So in this, one more concept I want you to learn. So there are two terms. One is the knockout and another one is knockdown. So these are not similar, these are different terms. So when we say gene knockout, so we means that gene knockout is an operation of genes. That means removal of genes from the DNA of an organism through different genetic techniques. So knockout means you are removing the entire genes from the DNA sequence. But what does gene knockdown means? So gene knockdown is an experimental procedure where the expression of a particular gene or genes of an organism is suppressed, reduced, or silenced. So whenever we are trying to silence the expression of the gene by the effect of some other agents or the proteins or some experimental procedure, so that is known as knockdown. But knockout means knock out. So that means you are just removing that gene from the organism. Then gene knockout facilitates the study the function of genes by analyzing the results of lost genes. So by performing the knockout thing, so you can study that what is the effect of that knockout occurring at the level of proteins. So this is the advantage of gene knockout. So now let's see the application of gene knockdown. So gene knockdown facilitates the study of oncogenes, genes that cause neurological diseases, viral infections, hereditary diseases, etc. So by performing gene knockdown, you can see you can study different oncogenes. So oncogenes are the genes that are causing cancer, whereas the genes that are causing neurological disease or viral infections. So these are the different methods which you use depending on the what type of study you want to perform. So there are two terms, knockout as well as knockdown, and this is the difference between both of them. So now let's move to the another question, which says the degree to which an allele of one genetic variant is inherited or correlated with an allele of a nearby genetic variant within a given population is known as. So now you have to tell that what.
Option A.
Okay, option
Option
A.
A.
Okay, so the correct answer will be option A, that is linkage disequilibrium. So I hope you all know about this. What does linkage disequilibrium means? And what is linkage equilibrium? Okay, so let's see. So in this, I've given you the two examples. One is linkage equilibrium and one is linkage disequilibrium. So whenever two genes are present, so in certain cases, these two genes are either linked and they are inherited in the similar way in the offsprings as well. So when there is linkage equilibrium, that means there is equal probability that both of these alleles can recombine from both of the parents. So here you can see there are one allele is capital A, capital B, and another is small a, small b. So there is equal chance that capital A can combine with capital B as well as capital A can also combine with small b. And similarly, small a can combine with capital B and small a can also combine with small b. So in this way, there is equal probability that you can observe these two genes and two different allele forms of a particular gene together. But when you talk about the linkage disequilibrium, so that means here you can see that capital A is only occurring with capital B and it is not combining with the small b, and similarly for the small a also. So in this, the probability of observing capital A and capital B is 0.5, and similarly for small a and small b is 0.5. But there is the probability of observing capital A with small b, its probability is 0, and similarly for small a to capital B is also 0. So that means their inheritance is linked to one another. So in this way, that is known as linkage disequilibrium. That means one particular allele will decide that the another allele will be inherited or not. So the expression of both the alleles is correlated to one another. So that is linkage disequilibrium. So hence option A is the correct option for this. So now let's move to the another question which says genetic disorders with anticipation. So these are the options. involves repeat expansion, affect both sexes, show severity of the disease inversely correlated with age at the onset in successive generation, or all of these.
All of
Okay,
All of this.
All of this.
okay, I hope you all are aware about the anticipation now. So yeah, correct answer will be all of these because in anticipation it involves repeat expansions. It can affect both the sexes and it also shows that severity is inversely correlated with the age at the onset in successive generations, as we have discussed in the beginning that how the repeats are expanding and how it affects. So yeah, the correct option will be all of these. Now let's move to the another question. So which one of the following options is a prerequisite for the generation of knockout mice? So as we have seen in the previous to previous question that knockout mice models are used to study different types of things.
Option
So
A.
okay, so option A is embryonic stem cells. Option B is knowledge of the cellular functions of the gene being targeted. Option C is Cre recombinase transgenic mice. And option D is all of these. So now here is you have to generate the knockout mice. So what is the prerequisite? So somebody else wants to guess? Okay. So somebody has answered embryonic stem cells. So yeah, it is the correct option. So for generating the knockout mice, the prerequisite is the embryonic stem cells. So here I've shown you the knockout mice that how it is generated. So it is a blastocyst and these are the inner cell mass. So from here the mouse embryonic stem cells genetically engineered to disrupt the gene. So in this the gene is being disrupted, it is removed as it is a knockout mice. Now this blastocyst is transferred into the uterus of host female where it will develop and now mouse containing a mixture of genetically engineered cells and the inner cell mass. So in this way, knockout mice is generated by using the embryonic stem cells. So option A is the correct option that for generating the knockout mice models, the embryonic stem cells are the prerequisite. So now let's move to the
but
another.
Cree
Okay,
recombination was required now, ma'am.
sorry, can you repeat?
But Cree recombination also required now.
Yeah, but here they are saying the prerequisite for generation. And when you talk about the Cre recombinase transgenic mice, so Cre recombinase is used when you want to observe the cell-specific or tissue-specific expression of a particular gene. So before that, if you want to generate a knockout mice, then generally the prerequisite is embryonic stem cells. And that Cre
Okay.
recombinase is used when you want to observe, means you want to study a cell-specific medium. Means in this you are studying for the entire model, but here if tissue your interest is to observe the expression of tissue specific gene, then you perform this Cre recombinase. So if we talk about the generation of knockout mice, so to generate it we use embryonic stem cells and then only we can knock out that gene. In Cre recombinase, after the sequence is present, after that you use the Cre recombinase mechanism to knock down that gene.
Okay, ma'am. Thank you.
Yeah. So now let's move to the question number 14. So to comprehend the mechanism underlying human disorders, model systems are frequently used because. So as we have discussed about the model system, so here you have to tell there are different mechanisms listed below. So why do we use the model systems? So option A is model systems can be utilized to verify whether or not the symptoms being observed are indeed the result of alterations in a particular gene sequence. It is possible to modify the genomes of the model systems so as to produce genetic abnormalities similar to those present in the human individuals. Option C is when compared to human individuals, studying changes in the developmental processes or cellular physiology can be done with a great deal more ease in the model systems. And option D is all of these. So you have to tell that why do we use model systems when we want to comprehend the mechanisms that involve human disorders and why we don't go for directly using the human individuals as it is. So what do you think? How these model systems make our life easier? So actually, there is one issue in this particular bullet is by mistake printed. So these are the four options. This is option A, this is option B, this is option C, and this is option D. So somebody is saying option
All of this.
all of these, right? So now let's see. So the correct answer is option D, all of these, because the model systems can be utilized to verify whether or not the symptoms being observed as a result of alterations in a particular gene sequence. That's why you use the knockout mice models. It is possible to modify the genomes of the model systems so as to produce the genetic abnormalities similar to those present in the human individuals. Because these model organisms show similar patterns of expressions as compared to humans, so that's why we use them. So similarly, by knocking a particular gene, we can see what is the effect in that model organism so that we can kind of relate or we can simulate the similar situations in the model organisms as present in the human to visualize the effects. And we can also design different, treatment methods, and we can try or test them in the model organisms. So when we look for the drug trials, so the trials, these are run on the model organisms only, and then these are further tested on the human individuals. And then, when compared to human individuals, studying the changes in developmental processes or cellular physiology can be done with a great deal of ease in the model systems. So, these are the reasons that why we choose the model organisms as compared to choosing the humans itself. So, these are the advantage of choosing the model organisms. So, this was all about the content and the MCQs that were related to the week 3. And in the next we will be discussing about the week 4 content. But if you have any doubts related to the week 3, then you can ask me. And if you have any doubts related to any of the questions, because somehow I am done with the content very fast.
Regarding the balance translocation, if you can explain in case of a disease scenario, it's quite less understood.
So, in with regard to which scenario?
Disease scenario.
Okay, disease. So, if we talk about the translocation that is imbalanced, so in Philadelphia chromosome also, if you see the translocation is imbalanced only because uh, a larger part of the single, so the two chromosomes are involved in which the translocation is also the imbalance. So larger part of one chromosome is being shifted to the another chromosome that is extremely small, the chromosome 22, and the chromosome 9 that is quite large. So imbalanced translocation, whenever it occurs, it leads to some kind of the disorder. It can lead to gain of function. It can also cause loss of function because the size of chromosome means there is something to do with the size. If a particular chromosome has, so it has two sister chromatids. If they are imbalanced, then it will there will be change in the sequence if there is imbalanced translocation, and hence further when it will code for certain proteins, then also there will be issue. So imbalanced translocation can also be allelic as well as non-allelic. So uh, means it can be the result of non-allelic translocation as well, in which there is no allele is being transferred to the another, but some other part is being transferred. So whenever there is imbalanced translocation, then it will always lead to some kind of complications in the chromosomes and further in the individuals in which the imbalanced translocation is occurring. So one such example is for the Philadelphia, in which there is gain of function, two genes, so one gene that was already present in that chromosome, so another gene is also due to translocation, another gene is also translocated. So these are a few examples of imbalanced translocations and how they leads to the disease because there are two sister chromatids that are joined together. So there will be imbalance. So further, when it will be passed on, then it will lead to the altered protein expression, and that protein can be further the cause behind the means that can further lead to the cancers or different disorders. So in that case, Philadelphia also it leads to the cancer because the new protein that is being synthesized that leads to the indefinite cell divisions. So um, So is your doubt clear, or you have some other doubt as well?
Yeah, but how it is different from like I have doubt regarding the
Hmm.
balance translocation. How does it affect any individual? And like that, you have given example of Philadelphia chromosome. What are the other examples could be found in case of balance translocation?
So in balanced translocation, so when we talk about the balanced translocation, then balanced translocation is normally observed when you talk about the recombination also. That is also the kind of translocation only of alleles from one homologous chromosome to the another homologous chromosome. So means if it is balanced, that means even after translocation, both of the lengths of the homologous chromosome is being balanced. And not just homologous, but the translocation can occur between the non-homologous chromosomes as well. So balanced means whenever the similar length. So for its, if you want to look into the examples, then I could not recall example as of now, but balanced translocation can also lead to disease. And it is not always that if it is balanced, then it will not cause any disease. So during balanced translocation also, it can cause some complications. So maybe for the next session, I'll find out certain examples related to the balanced translocations. But as of now, I can't give you the example for the balanced translocation. But yeah, balanced translocation can also have the detrimental effects on the individuals and means similar to the imbalanced translocations, depending on the
So,
region where the translocation is occurring and what is the significance of that region.
so does the balance translocation affect the gene expression of that gene where it is being translocated, balance translocated?
Yeah, it can affect. So balanced translocation can also affect the expression of the particular gene, that how it is being expressed. Because translocation, if we talk about translocation, that means the region between the two chromosomes is getting translocated from one side to the another side. So if there is some difference in both of the chromosomes that are being sharing that particular region that is undergoing the translocation, then it will have some influence on the expression.
Means I think the balance translocation could be good or could be bad, or it is always known to cause disease, or it is something like
Yeah,
that,
means
or something.
yeah, it can have effect. It can lead to some disease. It is not always that it is balanced, so it will not cause any disease, or means it will always cause disease.
So
So
in case of imbalance translocation of 9 and 22 Philadelphia chromosome, so
hmm,
it will always give rise to diseases like chronic myelogenous leukemia, right?
yeah,
So in case of balance translocation, so there's a doubt that whether is there any possibility that it will give rise to any disease, or it's like that it might or might not give rise to disease. What's that?
yeah. So it might or might not give rise to disease. So balanced translocations might or might not can give rise. But imbalanced, as it is causing the imbalance, so it will always lead to a disease or some effect at the level of the gene expression. But in the balanced, it can either cause some disease, it can be detrimental, or it can also be, it means it cannot have any effect if we talk about the balanced translocations. I will check for the few examples for the balanced translocation if there are present.
Okay. And one more is that the Angelman and the Prader-Willi syndrome.
Yeah, Prader-Willi and Angel.
Uh, can you help us understand using a pictorial diagram of the Angelman as well as the Prader-Willi syndrome? So in the theory, these are all imprinting disorders, right?
Yeah,
So
yeah.
how it is different from the other disorder? It will be very much helpful if you can use a pedigree to help us understand these two disorders.
Okay, so I think in my week 2 or in my week 1 session, I was discussing about the Prader-Willi and Angelman, that how the particular gene is being silenced. So in that, I guess I have not given information about the Angelman, but I have discussed the Prader-Willi syndrome. I'll check if I find some diagram related to the Prader-Willi. So you want to see the karyotype, that how the karyotype is different between both of them,
No,
or
the
you want to
linkage
see
analysis,
the
of these two disorders.
okay, so means how one copy of the parental gene is being silenced in one of
Like the pedigree problems, we usually solve like that. If it would be given, then it will be extremely helpful for us to understand. I think visualizing the things will give us a better idea how it is being done or how it is usually takes place.
okay, let me check. So the figure which I have here is about the regions or the genes that get imprinted or that get silenced. I need to find. So you can also tell me the other doubts or other topics that are that you want to know, so that I can prepare it for the next time, so I can keep those things in mind, and I'll keep their pictorial representation or other kind of representations. So that means in the class I don't have to search, because it's very difficult to find it on the spot.
Okay, and one more doubt is there if it is, I think, let it be solved in the next week. Okay, but still, we have doubt. Means I feel personally that in case of a disease gene,
Hmm.
uh, is it the fact that genomic DNA and RNA can be used to identify the genetic, the code, the coding region of any gene, or it is always the protein and the genomic DNA are used for determining the coding sequence of if a disease gene?
Coding sequence of the disease gene, and your question is that either the DNA or protein sequence is used to identify that.
Yeah, means sequence of a sequence. If we have the sequence of RNA, DNA, as well as
Yeah.
protein, right?
Hmm.
So, like I don't know exactly. I have confusion that protein itself, can it predict the coding sequence, or uh, it is
Okay,
associated
so
like the we
yeah,
see
you
the
are
coding
confused
sequence.
when we talk about the mutations, that there are some silent mutations that only occurs at the level of DNA and that are not observed at the level of proteins.
Suppose we start with the coding sequence. We need to know the coding sequence, right, of any gene, and we have sequence of what protein. We have collected the protein as well
Hmm.
as the protein's DNA sequence as well as the RNA sequence, right? From like it's associated the protein of the protein. So which the sequence would be helpful for us to determine the coding sequence accurately, or using a single biomolecule like protein or DNA, or it is always being done in a series, all the three sequences required.
So when you want to look at the level of functional significance, so mostly then we go for the protein level, because means if a particular means change in the sequence, if it is not causing any change in the amino acid sequence and the protein is being retained, because the function or the phenotype is observed when you look at the protein functionality. But in certain disease or certain cases, a protein is not formed. So if protein is not formed, then you have to look at the level of DNA or RNA, because in certain cases, what you find is that the expression of the gene is being silenced. The gene is not getting transcribed. So in that case, RNA will not be observed. To study that kind of disease, you have to look at the level of the DNA sequence. So it depends that to which
But
level that particular disease is associated. Means it is occurring at the level of gene, whether its protein is being expressed. For example, there are certain genes in which protein is expressed, but that protein is not able to perform its function. So then we can compare the protein sequences. But if particular protein is not formed, there is loss of function. So in that case, we look at the level of the matlab most the level which can give you the most in-depth details about the functions, about the expression, that is the DNA sequence. But as we know that in DNA it also contains the introns, and so mostly we focus on the RNA techniques, the RNA-seq studies. Whenever we perform any disease analysis, then most of the analyses are performed at the level of RNA. But people also look at the level of protein because they believe that not all the RNAs are going to translate. So then people just means to they experimentally check the means which RNA is getting expressed by performing the mass specs and all that. So in disease, we can see that there are different means mass spectrometry peptides that are generated for disease, and then RNA-seq studies are also performed. So these are the two major biomolecules that are studied extensively to look into the disease scenarios, that how the disease is being occurred as compared to DNA. So mostly earlier focus was on the protein, but due to the recent means developments, the major focus is on the RNA sequence.
so protein along with the RNA, we'll just look in order to determine a disease gene or to find out if it is a disease gene or not.
Yeah. So mostly those two things are majorly focused as compared to DNA
Okay.
because since it does not always
Mm.
contain the coding part. So
And for the linkage analysis, do we still rely on the technique of linkage analysis for determining any disease gene? Is it a, I think it's a forward genetic approach, isn't it?
yeah, so you can do linkage analysis as well if whether the particular genes are either linked to one another or either means that depends on the distance of the two genes and how they are correlating to each other's expression. So in those scenarios, you perform the linkage analysis. But if a particular single gene is independently causing some disease, then in that case, the linkage analysis, I don't feel it would be helpful.
Yeah.
So is my screen visible?
Yes. You're visible.
I've looked into the examples of the Prader-Willi and the Angelman syndrome scenarios. So I could not find that um,
Uh,
there.
can you please include few examples? I think the pedigree analysis and problems related to that, especially for the Prader-Willi and the Angelman. So these are the most hardest problem which I have seen or faced. So every pedigree analysis problem could be solved with a little bit of smartness, but these are the things which require something more than smartness.
Yeah, so in imprintings, yeah, there is means difficulty in solving because in most of the cases you'll see that the imprinted organism is not showing the phenotype, but still it is carrying the genotype. So
So, so my question is there any resource we can go through to help us understand this pedigree analysis better and we get to solve some problems as well? Can you refer any website or any, I don't know, any
so
resource so that we can go through and check out and can solve pedigree analysis regarding that?
actually one single resource I'm also not aware of a particular resource which you can rely on, but if you could see the web, you can find multiple problems related to that. Since as of now, I can't suggest any one of the major sites that you can go and study these. But I think if you find it means if you Google it, then multiple examples you can see there are multiple sites means there are different sites that requires you to subscribe to check the results, but still there are certain sites that provide you free of course. There are different universities also because when I was looking into the problems, then I saw that there are many universities which provides you with the different assignment questions and along with their solutions. So you can look into those also different universities. foreign universities that provide you practice problems. So you can type it online and you can see that there will be different. And if you won't be able to find, then in the next session, I'll check for different sites where you can find these problems. And in next session, I will also include few pedigree problems related to these. So as of now, I've just shown you the means how the deletion is happening in the Prader-Willi and the Angelman syndrome, which of the copy of the parent, which copy of parental chromosome is being silenced in both of these cases. And I hope means this is quite normal or means this is basic understanding for the Prader-Willi. I think the problems which you want to see is related to the pedigree analysis, right? For the Prader-Willi and Angelman, or
Yeah, yeah. I think
was this was
in
your
case,
question?
so in case of pedigree analysis, if we have to solve any question regarding pedigree analysis in the exam, so how we do it in an accurate way?
So, uh, in the week two assignment, have you come across that question in which they have asked you the minimum number of offsprings that shows the means that shows the genotype but not the phenotype? So that was related
Genotype,
to the imprinting disorder.
maybe, yeah, yeah, I have solved each and every question regarding the pedigree, thankfully, but
Hmm.
the theory part was quite confused, and I had done a few mistakes regarding that.
Okay,
I think,
so your doubt is specifically for the Prader-Willi and Angelman, or your
yeah,
doubt
yeah,
is
imprinting
general?
disorders, not regarding the theory, but practically how to solve the pedigree analysis regarding these two disorders.
Okay, so I will try to incorporate them. So in this figure, I've shown that which particular part of the in which chromosome is deleted in this case. So in this
Yeah.
in the Prader-Willi as well as in the Angelman, they have shown that for the in the paternal copy, this particular part here is being deleted. That is 15q13 region, and the paternal chromosome is deleted. Whereas in maternal, it is being retained. So due to which there is imprinting defects, and there is disomy of the maternal chromosome, and this part is being deleted, which leads to
So in
Prader-Willi.
case of Prader-Willi, is it a dominant or recessive disorder?
Prader-Willi, I guess it is the autosomal dominant disorder because when we talk about the imprinting, then genomic imprinting is observed in the autosomal dominant inheritance pattern. So I guess it is autosomal dominant.
So these two, whether it is Prader-Willi or the Angelman, these are considered as autosomal dominant, right?
Yeah.
The same as anticipation, like the trinucleotide repeats. So those are also autosomal dominant, right? With
So they are
varying in penetrance.
so as when we were discussing in the beginning, then we discussed that these disorders means these repeat expansion, they are not always autosomal dominant. They can be autosomal recessive. They can be X-linked recessive or X-linked dominant. So they can be means there are different modes of inheritance patterns that we can observe for these anticipation events in the dynamic mutation when we were discussing. So those are
And
not
like for
specifically
calculation of penetrance, we always see the autosomal dominant trend, right?
right.
The yeah, yeah. I got confused a little bit regarding the imprinting. The imprinting disorders is the same. It seems to be the most difficult part to be understood. Otherwise, we can understand, but this is the most difficult thing.
Okay, so I'll look for certain problem questions that are related to the genomic imprinting if I could find.
Okay, thanks.
Okay.
Thanks for the help.
So one
Thanks a
is
lot.
yeah, it's okay. So um, any other doubt anyone has? So you can ask.
Yes, ma'am.
Okay.
Ma'am, what is the difference between the trans-splicing and alternative splicing?
Okay. So wait a minute. I'll just alternative splicing I've already defined, but for the trans splicing I'll take one figure if I have. So wait for the trans. and cis splicing, there are two different terms.
Ma'am, because in that
Yeah.
question I was a little bit confused. I was thinking for the trans splicing answer, but the correct answer was the alternative slicing. So my doubt was ki, like why it can't be the trans splicing in the question.
Okay, just give me a moment. I'll find that question. Okay.
Question number 2,
Yeah.
I guess.
Got it. I was just looking for the image for the trans splicing so that you can see. So is the screen visible?
Yes, ma'am.
Yes.
Yeah. So this is the first one is showing the trans splicing. So there are three exons, exon X, Y, Z, and then exon. That another transcript there is exon X bar, exon Y bar, exon Z bar. So in trans splicing, what is happening is that exon Y is being replaced with the exon Y bar. Means the you know both these exons at the particular site is means that is trans. So at the same site they are present, but in this exon is means this exon Y is replaced by the exon Y bar, and then exon Z is present. But when we talk about the this kind of alternative splicing, then it is not defined that what exon pairs will recombine. So in this exon Y is combining means exon Y is X is combining with Y bar, and then exon Z is present. So a single exon is being replaced. But in alternative splicing, not just the exons will get means exchanged, but in other cases, exon can also get skipped. So when we say trans splicing, so in these two transcripts, we can compare them and we can say there is trans splicing due to which uh, this exon has shifted with that exon. But in alternative splicing, these two exons are present alternate position to one another. They are present in the alternate positions, and now in one transcript, one of the exon is present, and in the another, other exon is present. But here you can see all the three exons were different from these three exons in the another transcript, and only a single exon is being spliced and being exchanged. So that is trans splicing. But here there was a single pre-mRNA and different combination of exons combined together. So exon first and last exon is similar in both, but the middle exon is different. But here exon beginning and the both of them in both of the transcripts, beginning and the ending exons are also different. So that is trans-splicing. If we compare the two transcripts, then we can define the trans-splicing. But alternative splicing, it is defined for the entire transcripts that are being generated. And in the alternative splicing, it is not always the case that both the exons should lie adjacent to one another. There can be exon skipping events that in one transcript, four exons could be present, in another three are only present. Or there could be events where the intron is retained due to the missing of the splicing of that intron. The two exons get combined together. So that case is also you can observe in the alternative splicing, but not in the trans splicing. So the trans splicing is this, where the means single exon is being different in
Yes.
both of the transcripts, and it is defined when we compare the two transcript pairs. But when we talk about in the journal terms, then alternative splicing is a means broader term. It is not just for the comparisons.
Okay, ma'am, I got. Thank you, ma'am.
So as per Wikipedia, they define the transsplicing as a special form of RNA processing where the exons from two different primary RNA transcripts they are joined end to end and ligated. Okay.
Ma'am,
Yeah.
ma'am, I had doubt in assignment 3, ma'am.
Okay.
Second question, I think.
Okay. So in assignment 3, which question?
Second question, ma'am.
Second question. Okay, so what was that?
Sanger sequencing, ma'am. About Sanger sequencing.
Okay, so what was your doubt in that?
Ma'am, they have give something as copied DNA. What does mean by that, ma'am?
Copy DNA or cDNA?
Both, ma'am. Both. They are gave in bracket that it is cDNA, and
cDNA,
they
right?
broadly give us copied DNA.
Okay, so I'll try to share that with you. Wait. So is it visible?
Yes, ma'am.
Yeah. So this was your doubt. So in this, they have given the following schematic represents a DNA sequencing gel resulting from the Sanger method, assuming that the DNA strand being sequenced is the copied DNA. Identify the option that represents the mRNA sequence. Okay, so you have to tell about the mRNA sequence. Um, so what was difficulty that you found?
What is copied DNA, ma'am?
Okay, so copied DNA or cDNA. So in this, they have written it as a copied DNA. They have given the full form. So cDNA is a DNA. Whenever a DNA converts to the mRNA, so what happens is the introns after the splicing introns have been removed. Now you have got a spliced mRNA. So whenever this spliced mRNA is further reverse transcribed to the DNA, then that DNA is known as cDNA.
Then it will be a template strand, ma'am.
So template strand is the strand that codes for the RNA. So RNA is complementary to the template strand, right?
Yes, ma'am.
So
Uh,
RNA is exactly similar to the antisense strand, sense strand that is complementary to the template strand, the DNA strand. So that is the that will be equal to the cDNA, but in the cDNA, introns will not be present as they were present in the DNA. So its correct option was this 5' to 3' CCUAGC. In this, they have given that the sequence of the template RNA for the cDNA being sequenced would be reverse complementary to the sequence obtained from Sanger sequencing. Okay, so when you perform the Sanger sequencing, you get the sequence from 5' to 3' direction. So in this way, they are saying that that particular sequence from 5' to 3', that is the template strand, and then the cDNA will be reverse complementary to that sequence. So based on that, they have given you it from the 5' because it's from its 3' the sequence will begin. So that complement for the complementary. So if we convert this particular gel into a 5' to 3' sequence of the DNA, then you will see that it will code for the A, then AGT, CCA, TGC, sorry, GTC. So if you look from the top, then that is 3' to 5' direction code, that is GGT, GGAT. And you will see that when you convert into the, so from 3' to 5', you convert it into the RNA. Since it's complementary if you form, then that will be CCU because it is cDNA and it is get means you find you have to represent the mRNA sequence. So in mRNA there will be U in place of T. So that's why it will be CC in place of A it will be U. So CCU, then again U, sorry, then again A because T A is complementary to T, and then for here C is present. So in the complements RNA there will be G, then C, then A, then U. In place of A, so where in this band A you will observe, so its complementary will be T in the DNA, and for RNA it will be U. So in this sequence you'll find U here. So with
okay.
yeah, by looking at that, you will be able to figure out it's why this is the sequence. Because firstly, you have to write its sequence in 5' to 3' direction. Then you have to reverse from means because whenever you look for the transcription, then it always begin from the 5' to 3'. But the strand that is means that you choose that should be from 3' to 5'. So then you will be able to convert into the mRNA. So from reverse direction, if you will write its means sequence, then you'll be able to get this.
Okay, ma'am. Okay, got it.
Yeah.
Thank
So
you.
were there any doubt in this assignment part? In any other question?
Yeah, I think regarding the prerequisite, there was one question where they asked
Yeah,
what are the
you earlier asked
prerequisites? What are the prerequisites for uh,
this one question, right?
knockout
Generation of knockout mice.
mice? Yeah, yeah. So one doubt I have, brother,
Okay.
but if we do not know the gene, the function of the gene, what we are deleting, whether through the knockout, such global knockout or the conditional knockout,
Hmm.
and use of genomic library and use of flanking sequence and to include our gene of interest and to replace it with the neomycin gene, right? So how is it not correct that we first need the information of the gene which we are deleting rather than the obvious thing which we always need, the embryonic stem cells, inner cell mass from blastocyst?
Yeah. So when we say this model organism, so we means that we have the information already that which of the particular gene. So before performing these knockout experiments or any experiments, we already have the information with us, and then we just try to confirm that what is happening, whether this gene is, if we will knock out that gene, then what is the influence on the organism? What is the influence at the level of protein expression? Either that gene was coding for something that was important for the survival, or that was giving out some kind of phenotype which is now missing because that gene is being knocked out. So for this, the prerequisite, or we can say we already have the data in hand about which gene is causing what. And then this is in the model organism, we just try to test it out that either this means what is our hypothesis is true or not. So means, yeah, we have the information. We are just not randomly deleting
Yeah,
something.
but yeah, because if it is not been clear that the information are already present with us, then it will be rather easier for us to answer this question. If it is not mentioned, then confusion starts creating in our mind. So we think
Hmm.
whether the information of the gene would be required first, or uh, or
Yeah,
or rather the embryonic stem cells, it would be required obviously.
so in the embryonic stem cells also they particularly disrupt certain genes when they say they require embryonic stem cells, because embryonic stem cells have the ability to divide into multiple lineages of cells. Those are the main cells that will decide that what different cells would be produced. But yeah, in certain scenarios, if you don't have the information about the gene involved in the disease, then we go by the hit and trial methods. We try to means disrupt different genes, and then we check that in which of the following we observe particular expression that is being relating to the expression that is observed in the disease. So yeah, in certain cases, means either you have the information related to the phenotype, what kind of phenotype you will observe. Means for any experiment, you must require that either you have the information about the beginning point or either the ending point, that what of the results are you expecting. Then you can see that means after disrupting what kind of gene you observe still the similar phenotype. Or either you should have information about the gene so that you can know that if we silence this gene, what is the phenotype that we will see. So in this case, means either one of information is being required. That either you must have idea about the result that you will observe, or either you have the idea about what is the main causal agent, and then you will see what effect will happen. Either it will lead to gain of function or loss of function. But for the Cre recombinase transgenic mice, must have the idea
No,
about a particular sequence of interest or
yeah.
gene of interest, and then you will specifically silence that part by adding the lock P signals.
So it is quite understood that that Cree-recombinant mice is not going to be the case, because
Hmm.
if they have said the Cree-recombinant, then we can think of the option. But if they have said for knockout mice
Yeah.
can be global as well as conditional.
Okay.
So in
Yes.
case of conditional, we need Cree-recombinance. So
Right.
that's quite clearly understood that this is not certainly the right
Yeah.
option.
This will not be the right option. Right. Okay. So in the chat, many people have said that can you please show all the questions or slides once again as I joined the session late? Yeah. So for the people who have joined the session late, I'll just show the slides from the beginning so that if you have missed out certain things, then we will also revise it. So let me share the slides. Okay, so do you want me to again go through the ins- from these mutations also, or you just want to look for the questions that were there? So the people who are writing in the chat can tell me that do you want me to explain them again or just the practice questions? What do you want to see? Okay, questions. Okay, then I'll move to the questions. So these questions, this question was related to the PCR. So let's once again go through all the questions quickly. So in this question, it was asked that PCR can be used for different, means which one of the following processes, in which the first option was cloning a segment of the known coding region of the chromosome, then cloning the coding region present in the processed mRNA, mutation detection, and all of these. So in this, they were asking that for which reason can you use the PCR? So the correct answer is all of these, because by using the PCR, that is polymerase chain reaction, you can clone any segment of interest, no matter it is coding part or it is non-coding part. So in this way, option A and B will be correct. Then comes mutation detection. So you can also detect the mutation because you are amplifying a particular region, and then you can see that if there would be some mutation, then you will be observe it in the later stages by performing different experiments for observing the polymorphisms or the RFLPs. So with that, you can also look for the mutations as well by using this PCR. So in this way, all of these will be the correct option for this. And other than these things, you can also you can also perform genotyping, cloning, mutation detection, sequencing, microarrays, forensics, and paternity testing. So these are the different applications of PCR. The next question was related to the so okay, so in this question, I've added one figure while explaining it to someone about the transplicing. So let me first remove it so that it won't affect the question. So that question was related to the alternative splicing, and in that a figure was given. So wait. Yeah, so this was the question in which they have said that which one of the following options correctly identifies the processes given in the schematic diagram. So in this diagram, they have given you the pre-mRNA sequence, and they said that there is splicing and two transcripts have been formed, and then there occurs translation and these two protein isoforms are observed. So now you have to tell that what kind of splicing has occurred here. It is trans splicing, alternative splicing, cell splicing, or tRNA splicing. So its correct answer is alternative splicing, because you can see that in both of these transcripts, alternate exons have been selected, or you can see different exon combinations are there. So in this case, both the exons are adjacent to one another, and in that way, these both have been joined to the exon 1 and 4th. This exon 2 and 3 alternatively joined to both of them, and then their translation leads to the formation of the protein isoforms. So this type of splicing is known as alternative splicing, where different exons combine in different combinations in two different transcripts. And there can be different, subtypes of alternative splicing. There could be skipping of exons, because when we talk about the trans splicing, so in trans splicing, the two exons from the two of the transcripts are joined end to end, means the one exon is being exchanged between both of those transcripts. So that is trans splicing. And in alternative splicing, there could be the cases where one exon gets skipped, means there could be difference in the two transcripts in the number of exons, because in trans splicing the exon number is still maintained in both the transcripts, but in alternative splicing you can see that in one transcript you observe 3 exons, and the other you can observe 4 exons. Or you can also see that there is, in certain cases, introns could also be retained due to the uh, missing of the splice site. Spliceosome was not able to splice it out. So in that case, exon 2, 3 could be present along with the intron as a single exon in one transcript. So that is all is covered inside the alternative splicing. So the correct answer was alternative splicing. Then we discussed about the question number third, in which it was given that there are the two sequences. One is wild type allele, one is mutant allele, and this red one is showing the mutation that has occurred in the sequence. And this is the amino acid that is being coded from this sequence. And they've asked you that in the wild type allele, it was coding for histidine, cysteine, tyrosine, glycine. But in the mutant allele, you observe histidine, serine, tyrosine, and glycine. So what is this kind of mutation referred to as? So you have to tell what kind of mutation has occurred here. So as you can see that in both of them, if you see, the amino acid that was being coded has changed due to this particular mutation, that is a point mutation. So this kind of point mutation, where a change in the nucleotide leads to change in the sequence of amino acids, so that is known as missense mutation. So its correct answer was missense mutation. Then let's look into the question number fourth, in which they have talked about that you plan to develop a microsatellite marker. So in that, your means what you desire is that this one is the trinucleotide repeat and you want to expand it, you want to amplify it. So you have the information about the forward region to this as well as the backward region of this. So that means you have the information about the particular sequence that is present before this gene of or segment of interest which you want to amplify, and you also have the information about the sequence that is lying after this sequence of interest, and you have to perform PCR. So now you have to select one of the reverse and forward primers that would be the best pair which will help you to amplify these repeat regions. So that means these are the two sequences, 5 prime to 3 prime and 3 prime to 5 prime of the DNA sequence. So now you have to tell what forward primer you can use to amplify this. So as we know, the forward primer, it will bind to the region that is, as the name suggests, forward primer. That means it will synthesize in the forward direction and from 5 prime to 3 prime. So for a particular primer to replicate from 5 prime to 3 prime, you require a sequence of DNA that is lying from 3 prime to 5 prime. So that means forward primer will be used for this strand that is lying from 3 prime to 5 prime. So forward primer should be complementary to this region, that should be CAGTATGCA, that is present in the option number D. So this would be your forward primer. Similarly, if we talk about the reverse primer, so it will always, form, or it will always expand, it will always replicate in the reverse direction, that is from left towards right. So that means the strand which is 5 prime to 3 prime, for that we will use reverse primer that will lie in this reverse direction in this end. So its sequence from 5 prime to 3 prime will be from here. So that will be ATTACGCAT. So that is also present in this option D. So hence option D is the correct option for this question. So these forward and reverse primers would help you to amplify this region of interest. Okay, so now let's move to the next question. That was about the kind of mutation, what kind of mutation has occurred. So in this, they were saying that there is a steroid hormone receptor X, and upon binding to the steroid, it activates a set of genes which are involved in the physiological pathway which promotes the muscle growth. So due to genetic mutation, the receptor X was able to interact with few noble targets, and activate their signaling cascade. So which one of the following options correctly identify the type of mutation involved? So you have to tell that what type of mutation is involved. So in which you had a hormone receptor, and when it binds to steroid, then it activates the other set of genes. So here you can see that there is some kind of activation that has occurred upon binding. So whenever a certain binding or certain mutation, whenever it leads to the activation of other genes or activation of other pathways, then that is always the gain of function mutation. So in this, the altered gene product possesses a new molecular function or a new pattern of gene expression. So that's why its correct option or correct answer was gain of function mutation, because here they have already mentioned that it is activating other set of genes. So that's why its correct option is gain of function. Then we looked into the question number 6, in which they told that you have to tell the difference between the somatic and germline mutations, that how they are different from one another. So in the first option, they were saying that a somatic mutation is heritable, whereas a germline mutation cannot be inherited. Option B was a somatic mutation cannot be inherited, whereas a germline mutation can be inherited. Option C was a somatic mutation occurs in the DNA sequence, whereas a germline mutation takes place in the mRNA sequence. And the fourth option was a somatic mutation is deleterious, whereas a germline mutation is non-deleterious. So we have also discussed about this. So as the mutations which occurs in the somatic cells, they're not heritable because only the germ cells or sperms or egg, they are only, it means forwarding, or they are only passing to the next generations. So any mutations which are occurring in the somatic cells, they won't be inherited. Whereas when we talk about the germline mutations, so they are heritable mutations. So the correct answer was option 2, which says a somatic mutation cannot be inherited, whereas germline mutations can be inherited. Then we discussed question number 7, in which they have given you the agarose gel run after sequencing a DNA fragment, in which they have performed Sanger sequencing, and they have asked you to identify the correct sequence that would be generated. So in this, they have asked you to DNA sequence. But in your assignment number 3, they have asked you about the mRNA sequence, which was quite confusing for few of the students. But this was quite clear because in this, they only have asked you about the DNA sequence from 5' to 3' direction. So whenever we read a gel, we always start from the positive end because DNA moves from negative to positive. And whenever in the gel, whenever we perform electrophoresis, then one thing you must keep in mind that the fragments that are shorter in length will move faster in the gel compared to the fragments that are larger in length. So in this way, if you look from here, then the A will have moved faster and it will be the first fragment that is lying in the 5' end. So sequence will start from here. So this will be the shortest fragment. So 5' end will contain A, then C, then G. So in these options, you can see there is only a single option which gives you A at the 5' end. If we look from here, whereas in these options also, so it is quite confusing. So let's move to the other sequence after A. So then there will be C, then G, then 3C, then G, then 2A, then T, then A, then G, then again 3C, then A, G, A, and TT. So in this way, you will read this bands in the gel. So then you will get this sequence when you will read it. And when they have asked you about the mRNA sequence, so the mRNA sequence will be, or you can say the cDNA. So cDNA is nothing but cDNA is the DNA that you get from the mRNA. Whenever the mRNA is reverse transcribed to DNA, then it gives you the cDNA. And when we talk about the mRNA, so mRNA is formed from the template strand. And template strand is from the 3' to 5' direction, whereas the DNA which you get, mRNA which you get, that is exactly similar to the sense strand, that is complementary to the template strand, except for the T, you will see U in place of T. So in that way, you can find out the mRNA sequence for a particular given gel in which you have given the bands and then they asked you for the mRNA. So you can simply look for the 5' to 3' sequence, and from the 3' you can simply make its complementary pair, and when you will form its complementary strand, then in place of T you replace it with U. So in that way, you will be able to find the mRNA sequence of particular cDNA. So then we discussed the eighth question, in which they were saying that there is a mutation in a gene which is causing some severe neurological disorder in the offsprings of both the sexes, but when inherited through the mother and not when transmitted to the father. So in this, they are saying that a particular disorder is always means it is inherited from either always inherited from the mother and not from the father, which means that there is means silencing of the genes, sex-based silencing. So this property of mutation is likely due to. So what property of mutation is it? Is it complementation? Is genomic imprinting, or is it uniparental disomy, or is it anticipation? So out of these, we have already studied two different disorders that occurs that are observed when there is either one of the parent copy is silenced and only one copy is being expressed of the chromosome. So in that case, this type of mutation is known as genomic imprinting, in which either mother is giving the alleles or either father is giving the alleles. So it leads to the uniparental disomy. So uniparental disomy is the effect of genomic imprinting. So the property is genomic imprinting. So that's why its correct answer was genomic imprinting. So in this, I have shown that how the male and the female were given, and these are the alleles in which from the mother A was expressing and from the father B was expressing. So in this case, when there is androgenetic or paternal uniparental disomy, so there is no gene A, but there will be double dose of gene B. Whereas if there will be maternal uniparental disomy, so in this, father allele is being silenced, it is not inherited, whereas in mother, double dose of gene A will be present and no gene B. So this is known as genomic imprinting, in which only the one of the parent is able to express their allele. So in this case, the mother was carrying that neurological disorder gene. So whenever that particular allele is present, so particular offspring will show that disease, irrespective that is male or female. When we talk about the offsprings, but means the parent that is passing on is the mother. So even if that particular son is affected, but still that son will not able to pass it to the offspring. So now let's look for the another question. That was that the process marked as A in the image given below, which results in the generation of recombined chromosomes from the paternal and maternal chromosomes, is known as. So in this figure, they have given you the type of recombination, and you have to tell what kind of recombination event has occurred. These are the recombined chromosomes. So here you can see in the maternal copy, there is this allele. So both copy of alleles were shown in this color, whereas in the paternal, the two alleles were shown in this color. So in this, there is exchange of alleles. Here you can see in the offspring, there is recombination that has occurred. So one allele from the maternal and one allele from the paternal is being observed in the recombined chromosome. So this type of recombination event is termed as allelic recombination, and there is involvement of alleles that are being exchanged. And then I have given you the example where I've talked about the difference between the allelic and non-allelic recombinations and the consequences of non-allelic recombinations. So allelic recombinations, when the allele is involved and alleles are exchanged for one another. So as those are the alleles for the single gene that are being exchanged. So that is known as allelic recombinations. But when we talk about the non-allelic recombination, then what happens? The two homologous chromosomes are misaligned, due to which the different regions of the two chromosomes will be, crossovered or recombined with one another. So it can have the influence, it can lead to duplications in one chromosome and deletion in the another. So here these were the two alleles, A and B, that was similarly present in both of these homologous chromosomes. So due to misalignment, what happens is the in the first chromosome this particular part got deleted, so gene A and B got deleted from here, and they were passed on to this sister chromatid of this homologous chromosome. So there is duplication of this gene A and B in the one of the homologous chromosomes, whereas this gene A and B was deleted from in the another homologous chromosomes. So these are the consequences of non-allelic recombinations that further leads to certain disease scenarios. Then we discussed about the knockout mice models. So in this, the question was that for what of the reasons we can use these knockout mice models. So first was haploinsufficiency, Next was loss of function mutations, then was recessive disorders, and then all of these. So we use these mice models because they could help us to replicate the same situations in the mouse as they could be observed in the humans, and we cannot use humans because it was very difficult to observe, and it could be harmful also, and we could not be able to easily handle the human individuals also, and nobody would allow us to do that. But in mice, you know, people performs experiments, and the expression is almost similar in mouse also as they were observed in the humans. So different things that we can perform. So the correct answer was all of these because we can study haploinsufficiency. So I've told about the haploinsufficiency. Haploinsufficiency, it is combined by making it means it is a combination of haplo and insufficiency. It means that whenever a haploid copy is present, in certain cases, the haploid copy of gene is sufficient itself to form the protein that is required. But in certain cases, the single copy is not able to express the enough protein or enough gene, and due to which it is insufficient in its expression and it affects the individuals. So it is not able to maintain the dosage that is required. So that is known as haploinsufficiency. So knockout mice models can be used to study that. Then comes loss of function mutations. So we can also look for the loss of functions by studying the knockout mice, and we can also study the recessive disorders by looking at the knockout mice models. So in this way, we can use these models for all of these purposes. Then we discuss two terms, the difference between the knockout and knockdown. These are the two terms. So knockout. Knockout means when you knock certain gene out, you remove that from the organism. But knockdown means that gene is present, but by applying some external agents or some experimental procedures, you're trying to repress its expression. You are allowing it to repress and you are silencing it, due to which that gene is not expressing, but that gene is physically present. So that is the difference between the knockout and knockdown. So you perform the knockout when you want to study the function of genes by analyzing the results of the lost gene. When you want to analyze it, how the loss of that gene is influencing. But you perform knockdown when you want to study the oncogenes or you want to study the genes that are causing neurological disease, viral infection, and the hereditary diseases. So for those cases, you perform the knockdowns. Then we talked about the degree to which an allele of one genetic variant is inherited or correlated with an allele of the nearby genetic variant within a given population. So what is that known as? So whenever a single gene is influencing the inheritance pattern of the another gene, so there is a specific term that is known as linkage disequilibrium. So the correct answer was linkage disequilibrium. So what happens in linkage disequilibrium? That two alleles that are present nearby, they influence the inheritance of another, and they will always be inherited together in the offspring. They will never recombine with the another allele. For example, when we talk about the linkage equilibrium, then these two alleles are independent of one another, and from the other homologous chromosomes, they can recombine. So this capital A allele can recombine with the small b, as well as this capital B can recombine with the small a. So the probability of observing capital A, capital B, capital A, small a, capital B, and small b is 0.25. So there is equal probability of observing uh, these alleles together. But when we talk about linkage disequilibrium, that means the two genes are tightly linked and they will always be inherited together. And there is a formula to check that either two genes are in linkage disequilibrium or in linkage equilibrium. So whenever a particular alleles are in linkage disequilibrium, that means the probability of observing A and B together is 0.5 and small a and small b is 0.5. And the probability of observing capital A with small v is 0 and capital B with small a is also 0. So you will always observe them together and they will not recombine with the another alleles. So that is known as linkage disequilibrium. Then we talked about the anticipation. So genetic disorders with anticipation. So what does it involve? It involves repeat expansion, affect both sexes, show severity of the disease inversely correlated with the age at the onset in successive generation, and all of these. So the correct answer was all of these because we already discussed a lot about anticipation. So for example, in the Huntington's disorder disease also we have seen that how the trinucleotide repeats if that repeat is further expanded beyond a certain limit, then it influences the organism. So anticipation involves the repeat expansions, and it can affect both the sexes. It is not biased towards a single sex. And then it is inversely correlated with the age of onset, because with preceding generations, its onset will be at the smaller age. Because with each round of, replication, there is, means when we talk about the mechanism, so there is a slippage due to which more and more repeats are formed, more and more repeats will be formed, then it will further cause the effect. And with each round of replication, the repeat number will get increased. So means its onset will be at early age with the successive generations, and its severity is, directly correlated with each generation forward, the severity will increase. So all of these was the correct answer for this question. Then we talked about the knockout mice, that what is the prerequisite for generating a knockout mice. So its correct answer was embryonic stem cells, that why we require the embryonic stem cells and not the pre-recombinant transgenic mice, or knowledge of the cellular functions of the gene being targeted. So what happens in knockout mice? These from the blastocyst, these mouse embryonic stem cells are collected, and these are engineered to disrupt the gene. So in that gene is being disrupted. Now these blastocyst cells are transferred into the uterus of host female, and now the mouse contains the mixture of genetically engineered cells and inner cell mass. So now this is the knockout mice that have been generated. So these are the prerequisite for the generation of the knockout mice. Then we discussed the last question, that was to comprehend the mechanism underlying the human disorders, model systems are frequently used because. So in which we talked about the application of model systems, that why do we use. So there were different options which said that model systems can be utilized to verify whether or not the symptoms being observed are indeed the result of alterations in the particular gene sequence. Option 2 was it is possibly to modify the genomes of the model systems so as to produce genetic abnormalities similar to those present in human individuals. And then option C was when compared to human individuals, studying changes in the developmental processes or the cellular physiology can be done with a great deal more ease in model systems. Yeah, all of these. So its correct option was all of these, because model organisms make our life helpful, and we can observe different symptoms, different things by altering particular gene sequence, and they reproduce easily. They means we can observe the developmental processes and cellular physiology also with very ease as compared to the humans. So these model organisms are very helpful. And then somebody was asking about the Angelman and Prader-Willi syndrome. So these were the things in which one parental copy gets silenced, and then there is uniparental disomy. So in the Prader-Willi, only maternal chromosomes were able to express, whereas in the paternal, there is imprinting of this particular segment. And when we talk about the imprinting, then we must know that these are mostly the autosomal dominant inheritance patterns where this genomic imprinting is observed. So in the Angelman, the scenario is opposite, in which the maternal copy is being silenced, and the paternal copy maternal is silenced and the paternal copy is able to express itself. So this is the difference between the Prader-Willi and Angelman syndrome. And this was all about the questions and the concept which we discussed in today's session. So now is it clear? The people who joined late, so for them, is it clear? Do you have any questions or any queries? Or for the next session, you can also tell. So some students have recommended that for the next session, I should involve certain pedigree patterns that are showing the genomic imprinting kind of inheritance related to Prader-Willi and Angelman. So you can also suggest few topics so that I can involve them for the next session, which you want to understand, in which you find the difficulty related to any week. It's because in the next session will be our last session. So, okay, so if you have no further doubts, then I'll end the session. Okay, is it clear?
Yes, ma'am.
Okay, thank you. So I'll end the session, and next session I'll try to add few more questions, few more problems so that that session won't end so fast. Okay, thank you so much and see you in the next session.
Thanks a lot, ma'am.
Thank you.